Thanks, Harsha. I just want to congratulate you and the rest of the team here for this fantastic meeting and show. I think over the course of next 15 minutes or so, I will try and give you a few minutes of discovery activity. It's too sciencey, but I will try and make it accessible to everyone here. And also, I've been doing a lot of work in India, working across for the last almost 20 years, doing research work there, and also doing work in this part of the world. So I'm going to make a case, and I think it was pretty obvious with all the conversations that had happened, why India is a good place for rare disease and even drug development. But before I get into the talk, I work as a chief genomics and informatics scientist at MetGenome, and I run a company called Antler Therapeutics. It's the antibody-based regeneration medicine company, and we actually have our first drug in clinical trial. And this foundation, Genome Asia, was actually set up by MetGenome and many other colleagues to actually collect genomics data and to bridge the gap, and I will come back to that in a second. This is one of my sort of favorite slides in that it kind of gives you a flavor for India, the languages, the culture, the various aspects of it. And in fact, we saw the color yesterday at the evening celebrations. But this is a picture I took, probably only place in the world where they put an elephant in a truck and actually take it across the road. And it's fun sitting in the car watching this. And it is full of surprises. There is no dearth of very interesting things. Now, having said that, India's population structure, there is over 4,600 anthropological groups. I'm sure some of you heard Tangraj's talk yesterday. Would appreciate he and team have done a lot of work in the early days, along with Dave Reich, Harvard here. But this 4,600 anthropological groups basically also contains many tribes, and some are hunter-gatherer tribes. And the population group structures kind of evolved over 2,000 years, where the caste system and also the fact that people married within certain communities created a very unique but large population census that's very ideal for human genetics. And that's what New York Times wrote some time back. And the consequence of that is also a higher burden of recessive disorders, partly to do with ancestry and endogamic practices. There are 5 major language groups, 121 languages spoken, and each of these census group is greater than 10,000. And India's birth rate or number of births is 26 million live births. To give you context, it's the size of Australian population every year. So anything that's rare, considered rare, is really not rare, not because of incidence, but surely because of the size. And of course, today it's the most populous country in the world with 1.4 billion people. And when I was kind of thinking about India and trying to do something, going back and forth over the years, one obvious thing, which was my area of expertise and passion, genomics, where I've spent bulk of my time the last 20 years while I was at Genentech. If you look at the genomics data out there, there's this still missing diversity, as the aficionados call it. It's partly to do with still 98% of the data comes from people of European ancestry, partly because that's where the funding comes from. And so when you try to study disease-causing variants in other parts, and particularly in India, if you don't have the baseline variants, you're very likely to miscall things. So we set out to fix this. In 2011-12, we published the first female genome India out of genome from India. This was a cross-country collaboration between India and many colleagues here in the US. At that time, sequences one genome was almost $100,000. But we went ahead and did it partly because we wanted to kind of do something in that part of the world and prove that it could be done. And many colleagues provided annotation and a lot of support, and we ended up publishing it. And the heels of it, we were at a meeting called the Bones to Genes in Sitges in Spain. And out of this was born the Genome Asia Consortium. Partha Majumdar, who's at the right corner, he had founded an institute in Kalyani around genomics in India. But while he was at Indian Statistical Institute, it collected a large number of samples, amazingly with video consents and whatnot. So he was willing to contribute this for sequencing and to create a database of sequences from across major population groups in India. And that's what we did. This was a working meeting in Singapore. The consortium is based out of Singapore. And many groups, including other companies, participated in this. Out of it, we were able to document the largest number of variants from this part of the world that were not previously reported. And the rest is somewhat a history, but just to give you a flavor for it, that's the 5 language groups in 5 different colors that you see here. And we were able to survey and get baseline data. Still, it's not comprehensive to the extent we'd have liked, but it's a great start with over 1,000 genomes in there. And in fact, right off the bat, we could use this data to show we were studying early onset diabetes, maturity onset diabetes, MODY in young children in southern India. If you didn't filter with this data set to remove common variants, there were papers before that published a variant that had nothing to do with the disease because they didn't know it was common, and they called it a pathogenic variant, and in fact, it's not. On the other side, it also helps us clean up and actually find the right variants. This is a recent paper that we published. This is the second part of the Genome Asia Consortium, a very large amount of data, about 5,000 genomes. And we also built a imputation array. I know sequencing costs are coming down, but still an array at $40 is still attractive. And this has variants from that part of the world that allow you to get better imputation. And all of this data is publicly available. We have a browser set up a while back. And as we were doing this a number of years ago in collaboration with Dr. I.C. Verma, who was won the award yesterday, we started a study, a familial genetic study, to get this landscape of the variants within disease families with inherited disorders. We did not restrict to any particular disease. But interestingly, neurological disorders are the greatest, and parents really want to know why. And even if you don't have a solution, it's a closure at best for them to know what is going on. So out of this, again, we have a large number of variants and data collected. And I won't bore you with it, but I'll just show you a couple of vignettes. Some of this we've been publishing along the way. This is an inherited lung cancer, very, very rare. But in India, his character in Mumbai, his name is Dr. Kumar Prabhash. He called me and he wanted me to sequence a family of 3 people who, within a few years, they're in their 40s, got lung cancer. And he would bug me every time I see him in India. And I said, hey, this is so rare. I'm not sure what we're going to get out of it. But he convinced me in a moment of insanity, so to speak, but was the best thing. And we actually sequenced the 3 members in the family, or 2 of them, because one was already deceased, and a few others. And using this Genome Asia data set, we were able to filter all of the non-relevant variants. And my postdoc at the time came running to me and basically said, wow, we're finding a mutation in HER2, which is the favorite gene at Genentech, and for that matter, the first target on a cell surface that an antibody against which till today protects women with HER2 cancer in a remarkable way. And we knew this mutation is important, but activating a tyrosine kinase on the surface, it's a little bit, you'd think that you would not have a normal birth, but these people are born normally, they carry this variant. And as the structure of HER2, the extracellular domain and the kinase domain, and these tend to generally heterodimerize with other partners to activate signaling, particularly in cancer. And our thesis was this mutation leads to homodimerization. Since it's all published, I won't spend too much time other than to say we quickly assayed this protein. We put it into our cell system to show that it's highly active. I had shared this data with, sorry, my slides are not advancing for whatever reason. Sorry, I'm going to have to get out and restart it. Don't know why it's doing it. The wheel of death. Bear with me. Okay. See it. Asking me for feedback at the best moment possible. Right? Yeah. Oh, I know why this is not working. I should have taken a picture of this, but you'll have to believe me. This is actually, yeah, it's some refresh problem. So maybe there's actually 2 slices of lung CT scan from the patient, because I shared this data with Dr. Kumar Prabhash, and I hadn't heard from him in a while. And then excitedly he called me and said, oh, we treated the patient based on the mutation data with a drug that was available in India. Antibodies against HER2 are not available, or available but inaccessible, very expensive. So he treated them with lapatinib. And the patient had been given a notice, basically saying, give notice to your family. And she was labor, she couldn't breathe, very labored, and was bedridden. And after one course of the drug, she got a dramatic recovery. And she was back in the hospital and cheerful and walking around. And people got so excited that that's the treated side of the lung. Before treatment, there's a bigger mass. Anyway, this was one of these cases where we were actually quite delighted to see all of this coming together. Then this is another case. These are kinds of families that you can find. You may think this is a whole village, but it's just one family in Kerala where the family has this hypertrophic cardiomyopathy. The clinician who was seeing them again reached out and said, hey, can we sequence them? Of course, you can do very clever things and sequence index patients and do smaller sets of the others. And this is precisely what we did. In this condition, many people who suspected to have HCM, you give them actually, you put them on a pacemaker, but some of them wouldn't need it. Some of them would need it, but based on the clinical symptoms, the clinician decides to implant. But then he said, if we are able to sequence and there's a molecular basis for this, it'll be amazing. So, which is exactly what we did. And it turned out there were a few patients that they had misdiagnosed and wanted them on pacemaker, and the others who needed pacemaker but were not on the list. So we were able to actually address that. And this is a case of a familial pediatric cancer, very unfortunate. 2 kids, both of them get cancer. There's when we sequence, we found there's a mismatch repair gene mutation. And unfortunately, both parents are carriers. And both times when the kids were born, they were homozygous and they developed this childhood cancer. At least these parents were gratified to know what had happened, and I'm not sure if they did anything different after this in terms of having another child. The sort of the last example before I tell you a bit more a translational story. This is a very rare mutation in a kinase PRKG2. And this is found in a family where the affected individual has bone defects, not really short bone stature. And before this, when we found this, we were looking for what does this mean? It turns out the cow on your left, a dog, a mouse model of knockout, and also a rat that occurs naturally, all carry same mutation, the same gene. Again, telling you a little bit about the biology of what's going on here. So the next few minutes or so, I'm just checking time. Maybe I'll take 2 minutes and I'll try and wrap up. Is the translational story on how we've gotten a drug into retinopathy. This is to introduce Wnt signaling. It is an important pathway, very important for regeneration. The worm on the left can completely regenerate even if you cut it into multiple pieces and put it on the nutrient media. The Mexican salamander in the middle, cut its vertebra tail, it'll grow. But go to humans, we replace skin, gut, or corneal cells every day or every 3 to 4 days. Other organs, we have stem cells that allow repair and maintenance. And this is all fundamentally dependent on a single pathway. Of course, there are other collaborating pathways, but this is the most important one called the Wnt signaling. There are 2 receptors, frizzels and LRPs, a ligand called Wnt. Wnt comes in 19 flavors, frizzled in 10 flavors, and LRPs in 2. Combinatorially, the Wnt's activate, but they're very difficult to make as a ligand. You can't make it like insulin and use it for treatment. So we developed an antibody. This is actually an antibody that turns on the pathway, and that's what's shown in the bottom here. And how does it relate to retinopathies? Fairly straightforward, at least one indication. This is the fundus image in the inner retina showing the blood vessels as a cartoon. They're privileged. Means they're sealed. They don't leak any content. And that's dependent on this Wnt signaling pathway. And it maintains the cell-cell junctions of the endothelial cells. The minute the signaling is actually lost, you get massive leakage and you actually end up with retinopathies. There's pediatric rare diseases, nori, fever, coats, again, same mechanism, but not exact same genes. And also even diabetic retinopathy. So these are all the number of diseases where the biology speaks and we know the genetics behind it. There are real people affected by it. The Stevie Wonder or a young kid, a mom's trying to do something about this. And it's a disc jockey who has fever. And he's going through episodes of stuff. So we've been working in India actually to create a cohort and a small registry there. Out of that, we made our first drug that targets Frizzle4 and LRP5. This is a knockout mouse to which we give the drug. It actually restores the complete architecture. We've done a lot of work since then, all summarized in this single slide. This is our new format of the antibody. This is the one that's gone to clinical trials actually now. We have a partner with whom we work called iBio. I'm very grateful to them. And we have a number of pipeline candidates. I won't read the summary in the interest of time, but I just want to thank all of you for listening, and particularly all my colleagues at MetaGenome and my former colleagues. And I just want to leave a thought. I was earlier talking to Neil. We've been trying to make actually drugs in India, and the idea is why not east to west rather than west to east? Topic for another day. I'll stop there.